In this unit, we will discuss three key topics: the role of protein structure in determining protein function, the process of translating a specific messenger RNA sequence into its corresponding amino acid sequence, and the mechanism of translation in the cell. This video will focus on topic number 1, protein structure. Proteins, also called polypeptides, are linear polymers of amino acids that fold up on themselves to form three-dimensional structures with specific functions. Function arises from the shape and structure of the folded amino acid chain. There are 3 levels of protein folding that all proteins contain, and a 4th level found only in certain proteins. The first, or primary structure of a protein, is the sequence of the amino acids that are covalently linked together to form the polypeptide. The second involves localized folding of nearby amino acids. The 3rd level dictates the overall 3D shape of the protein, and the 4th level, found only in some proteins, is due to the interaction of more than one polypeptide chain, resulting in protein subunits. We will discuss each of the structural levels in detail. If you remember from Chapter 2, all amino acids contain an amino group, a carboxyl group, a hydrogen attached to an alpha carbon, while the R group varies between each of the 20 amino acids. A polypeptide is then built when each amino acid is covalently attached to another with a peptide bond between the carboxyl group of one amino acid and the amino group of another. This chain of covalently linked amino acids makes up the primary structure of the protein. The primary structure can be labeled using 3-letter abbreviations of the amino acid names. In this example, the amino acids in the primary structure are alanine, methionine, alanine, and methionine, which when abbreviated would be written as alanine, methionine, alanine, methionine. Based on the sequence of amino acids, which provides the order of the R groups, the linear primary structure will fold up on itself in a particular way due to the chemical interactions of the atoms that make up the amino acids. The first level of folding, called the secondary structure, occurs due to hydrogen bonding between atoms of the amino and carboxyl groups of the peptide backbone. Notice that the amino groups contain a nitrogen-hydrogen bond. Nitrogen is much more electronegative than hydrogen is, thus this is a polar bond, leaving hydrogen with a partial positive charge. The carboxyl group contains a carbon double bonded to an oxygen. This is also a polar bond, leaving oxygen with a partial negative charge. So the oxygen of the carboxyl groups can form hydrogen bonds with the hydrogen of the amino groups. These interactions cause localized folding in the polypeptide, resulting in the secondary structure. There are 2 types of secondary structures found in many proteins. The first is the alpha helix. This structure occurs when the hydrogen bonds between the carboxyl and amino groups form in a repeating pattern between every 4th amino acid in the chain, causing the chain to twist into a helical structure. For example, if we take this string of circles representing a chain of amino acids, this would be the primary structure of the protein. The secondary structure would form when small localized portion folds up into an alpha helix because of hydrogen bonds between every 4th amino acid, resulting in a structure that looks like this. Notice that the alpha helix only occurs in a small portion of the overall polypeptide chain. The second type of secondary structure is the beta sheet, which occurs when the polypeptide folds back and forth on itself, forming a pleated sheet that is stabilized by hydrogen bonds between carboxyl groups in one chain and amino groups in the other chain. The folded chains can be parallel, running in the same direction, or anti-parallel, as depicted by the arrows running in opposite directions in this figure. Looking at the secondary structure that we folded in the last slide, we can add a beta sheet to another localized folding area. Notice we will still contain our alpha helix, and then in another area of the protein, we will get a different round of localized folding, forming a beta sheet. Some proteins contain both alpha helices and beta sheets in their secondary structure, while others contain only one or the other. While the secondary structure of a protein results only in localized folding based on the interaction of the carboxyl and amino groups, the tertiary structure leads to the overall three-dimensional shape of the protein and is caused by interactions of amino acids in the R groups. Remember from Chapter 2 that the R group varies between each of the 20 amino acids. R groups allow amino acids to be grouped based on the chemistry of the atoms it contains. R groups can be described as hydrophobic or hydrophilic. Hydrophobic amino acids are nonpolar because they are composed mainly of carbon-hydrogen nonpolar bonds. Hydrophilic amino acids can be classified as polar if they contain polar covalent bonds, basic if they contain positively charged atoms, or acidic if they contain negatively charged atoms. These properties of the R groups are very important because they influence how a protein folds and its three-dimensional shape. Remember, shape dictates function, so the R groups have a major role in controlling the function of the overall protein. The amino acids shown here are considered hydrophobic amino acids. You will notice their R groups consist mostly of nonpolar covalent bonds. Hydrophobic amino acids tend to be buried in the interior of folded proteins because the hydrophobic R groups aggregate together to stay away from the aqueous environment of the cell. You do not need to memorize the structures of the amino acids. However, you do need to understand how the chemistry of their R groups affects protein folding. Hydrophobic amino acids are composed of amino acids with R groups that have either a partial charge due to the presence of polar covalent bonds with highly electronegative elements nitrogen or oxygen, or that have a full charge due to the acidic or basic nature of the group. The presence of charges allows the R group to interact with water molecules. Polar amino acids are also able to interact with other molecules through hydrogen bonding, while the basic and acidic amino acids are able to interact with each other or other charged molecules through ionic bonding, or the attraction of opposite charges. Overall, the hydrophilic R groups and their interactions cause the protein to fold in such a way that they are located towards the outside of the protein. There are a few amino acids that are noteworthy due to their effect on protein structure. Glycine's R group is a hydrogen. Thus, the central carbon atom is bonded to two hydrogen atoms, and glycine is not asymmetrical. It is nonpolar and small, which allows for easy rotation of the amino acid, increasing the flexibility of the polypeptide backbone. Proline's R group is covalently linked back to the amino group, causing an opposite effect. It causes the backbone to be more rigid and prevents protein folding in specific ways. The third amino acid is cysteine, which is the only to contain a sulfur. Two cysteines can form a bond between their R groups called a disulfide bond. This covalent linkage forms a cross bridge that can connect different parts of the same protein or different proteins. The tertiary structure forms because of the spatial distribution of hydrophilic and hydrophobic R groups along the molecule and the chemical bonds and interactions that form between those R groups. For example, if we look at our string of amino acids, which has been folded into secondary structures due to the hydrogen bonds between the atoms of the peptide backbone, the next step of folding, the tertiary structure, will occur due to the interactions of the R group. If this amino acid here is basic due to a positively charged R group, and this amino acid here is acidic due to a negatively charged R group, then the positive and negative charges can attract, causing the protein to fold like this. Here's our alpha helix from our secondary structure. Here's our amino acid with its positively charged R group. Which is now interacting with the negatively charged R group, causing our overall 3D shape to fold up into this structure. The tertiary structure occurs due to multiple R group interactions, such as ionic interactions, which we just demonstrated, hydrophobic interactions, dull sulfide linkages, or hydrogen bonds. There are 3 common ways to illustrate the tertiary structure of a protein. The first is the ball and stick model, which is useful to show the atoms of the amino acid chain. The second is the ribbon model. This is useful to show the secondary structure of the alpha helices and the beta sheets. The third is a space fill model, which shows the 3-dimensional shape and contour of the tertiary structure of the folded protein. Some proteins also have a 4th layer of structure that is necessary to their function. This is called quaternary structure and occurs when 2 polypeptides interact as subunits to make up a single protein. The 2 examples shown here show the different polypeptides in different colors. If you look closely, you'll notice that these polypeptides are not covalently bonded together. The quaternary structure is formed by non-covalent interactions, such as ionic and hydrogen bonds, that occur between amino acids of different subunits. Ultimately, the primary structure determines the secondary and tertiary structures of a protein, and the tertiary structure determines the protein's function. The contours and distribution of charge on the outside of the molecule and the presence of pockets, such as this one here, which might bind with smaller molecules, all play a role in the protein's function. Proteins can be denatured or unfolded by chemical treatment or high temperatures. When this occurs, they lose their function because they have lost their shape.